Enough. Not me. All right, let's get comfy. Mm. All right, let's give people a couple of minutes to roll in.
Hello. Don't forget that I'm on a Zoom call. So you need to tell her off quietly.
Sorry, my door's getting in the ear about something. All right, give it another minute, and then we'll pick up where we left off on Friday. Actually, this is probably a good time to ask if anyone has any questions about any of the material that we've covered so far. So I'll try and use the first, I don't know, however many minutes, 5, 10, 15 minutes of each Zoom session to go over any questions students have from the previous one, right? So we can use that as kind of like a catch-up, recap, you know, fix any problems that people might be having kind of deal. So does anybody have any questions about stuff that we cover? You don't have to. This will probably be more the case later on, but if you do, pardon me, now's a good time to ask. Blink, blink. Pardon me. All right. Well, if you do, just virtually stick your hand up. I guess there is a way of doing that now, which is kind of funky. Um, or type it in the chat box, or unmute your microphone and just ask. Any of those will work. Okay, so we have the rest of the flow of information to cover. Today we've got like a little bit of a leeway day today because we're not pegged to start Mendelian genetics till Wednesday, but we can start that, get an early go on it, I guess, if we finish the flow of information today, which we probably should, but we'll see how it goes. And as always, anytime you got any questions, just pipe up, right? That'll make things more varied and interesting. Other than the fact that it's a really good idea. Okay, so if anyone's got any other questions, I'm going to crack on. So let's do this. All right, I can't remember exactly where we were on Friday. I think that we were just finishing up the whole kind of overview of gene expression and about to start going in on translation, I think. Now, remember again, these topics are ones that we'll be going into in quite considerable depth later on in the course. So really, this is more of an overview kind of put stuff in context kind of deal rather than nitty-gritty details, because those will come later. But it is important to get the concepts, because otherwise those details are not going to make any sense. Right. Okay, so again, just to recap, essentially, gene expression is a means of getting the information that's encoded in DNA, which is all DNA is really, into a form where that produces something that does something, right? So typically as a protein, and proteins can be all kinds of things, right, in terms of their function. They can be structural, like actin or myosin. They can be motor proteins, right, which are particularly cool things like kinesins and dyneins, which literally walk along cytoskeletal filaments, like tiny little ropes essentially inside your cells. Quite considerable distances too. If you think about how long the maybe dendrites or axons, probably dendrites, that go from the base of your spine down to your toes, for example, right? Or even axons, depending on which way the information is going. They can be enzymes, obviously, that's something that you're a little bit more familiar with. Most likely they can be signaling molecules, right, which carry information of their own, right? So this isn't just the information of DNA, this might be information about physiological state or sensory information, all kinds of stuff, right? So, pardon me, the thing that is, in my mind, super cool about all of this is that DNA is made up of 4 bases, A, C, G, and T. RNA is also made up of 4 bases, A, C, G, and U. Protein's made up of, I always get this, I can never remember if it's 20 or 21. I think it's 20 amino acids. I should actually like just tattoo that on the back of my hand so I don't forget every semester. But those 20 amino acids are able to make over 20,000 different proteins, right? So our personal genome inside every one of our cells has information that encodes about 20,000 proteins, right? So I think that's just astonishing, right? Not in lots of different ways. How many different proteins we have? 20,000 is a huge number, right? It's kind of actually a bit beyond our ability to comprehend, I think. But also the fact that those 20,000 proteins, which have hugely diverse functions and shapes and all kinds of stuff, right, are all encoded by a form of information that uses 4 letters. I think that's pretty astonishing. And we'll see, how that works in a wee bit. Anyway, so the second part of this whole deal, right, transcription is obviously transcribing, right? That's kind of a neat word to use. That the information stored in DNA into a form of information that can leave the nucleus and then be used. And that process, that using that information in mRNA, is called translation, right? And so essentially we use 3 mRNAs and a couple of proteins, and actually a bunch of other proteins too, to translate that mRNA. We use our ribosomal RNA, which is the enzyme. We use tRNA, which is the translator, which brings the amino acids in. And the ribosome is the protein, or proteins, really, that house all of this stuff and hold everything in the right context so that can occur. And then at the end of it, we have a what we call the primary protein sequence, right, which then adopts various conformations and ends up as in its functional shape. So kind of bouncing around a little bit. I can't remember whether we covered this or not. If we have, give me a shout. Sometimes I get a little bit confused between my 2 classes, what I've covered in one and what I've covered in the other. Have we gone over this picture yet? I'm pretty sure I have for my other class. I'm not sure if I have for you lot. Speak now or forever hold your peace.
This is Amber speaking. I don't believe we've gotten this far.
Okay, awesome. Thank you, Amber. And whoever replied in the chat, where's the chat box? Ah, get up there. No, come on. Chat box is hiding. Sorry, whoever replied in chat, but thank you. Right, so this is actually a really cool image, right? It's really cool for a lot of different reasons. Obviously, it shows you the mRNA being created, right, being transcribed. So this kind of pale green ribbon here is the mRNA. And if you look at it, this actually looks very much like DNA, right? So if you look up here, this is our double helix. Down here, we also have a double helix, but it's made up of one strand of DNA, one strand of mRNA. Pardon me. So the same processes that apply to base pairing between DNA strands also applies to base pairing between mRNA and DNA, right? That's how we get the specificity of that transcription process, right? It's not just a random collection of RNA nucleotides, it's a very specific sequence, right? Another reason why this image is pretty cool is that you can see that the mRNA is growing, right? Hence the little arrow bit at the end, in a 5 to 3' direction, right? So just like DNA, mRNA can only be extended or grown, I guess one could put it, at the 3' end, right? So you can see if you like kind of lean in and squint a little bit, you can see that the next base, the next RNA nucleotide that's going to get added here is going to be oh, here's a question. What's it going to be? Question to the crowd. Let's see if I can bring up the chat box one last time. God dang it. This is like the most frustrating thing about Zoom is that it's very hard to see the chat when you've got your screen being shared. Can anybody guess or tell me, not just guess? There we go. Oh, that does it. Oh, there you go. Jimena's already answered that in the meantime while I was flapping around with this. Yeah, it's going to be a G, right? So you can predict what letters are going to be added based on this strand. Also shows you the whole anti-parallel deal, right? So the DNA strand is going down, right? It's kind of that's being copied, right? And so the RNA strand's going up. So they're also anti-parallel as well, right? And Jim, don't worry about being late. Just if you have any questions, just ask. What was I thinking about? Oh yeah, so this transcript's anti-parallel, it's going in the opposite direction. That means that, but we read the DNA in a 5 to 3' direction, right? So the mRNA being produced and the gene itself, right, are all in the same direction. So that means we have two different strands in DNA that have different functions. And this is something that's kind of a little bit ahead of where we're at, but it's kind of good to kind of plant this seed in your mind so that when we come to it later, it makes a little bit more sense. But this strand here, you see the one that doesn't have any mRNA attached to it, that's actually the coding strand, right? This is a strand that encodes the information that's being transcribed. So if you kind of go over, cross between these two, we've got a T over here, got a U, got an A, got a C over here, got a C, got a U, got a T, got a C, got a C, so on and so forth, right? You can do that yourself, right? So this strand here is the
Hey!
Puppies are fighting each other, making a lot of noise. I'll have to show you them one day. It's just a little distracting. So anyway, joys of teaching online from the couch, right? So this is the coding strand. This contains information. The other strand that's actually kind of base paired, that's serving as the template, is called the template strand, right? And so the two strands of DNA have very different purposes. This is the coding strand, contains the information that in the actual gene. This is the template strand, and that's the one that's going to have the mRNA transcript being copied from it. So it's actually, I love the whole field of scientific illustrations, right? Because science is it's full of facts and principles and concepts and stuff. But it's very hard to convey those concepts and the like by words. And so if you're able to be good at art and understand science, there's a fabulous job just waiting for you. And the people that do that are in very high demand. I actually spent a little while chatting with one at a conference once. Fascinating job. I can do it because I'm not much of an artist, but he did all kinds of cool illustrations for all kinds of different research groups and publications. Anyway, so basically the idea of gene expression is, we've got this whole transcription process going on, but also, and that's like the coding region of a gene, separate, well, in addition, not separate, in addition to that coding region, we also have what's called a regulatory region, right? And that's what controls whether or not that gene is expressed at any given point, right? Or context or cell or whatever, right? So really, when we talk about a gene as a unit of information, we're talking about both of those together. We're talking about the actual information, which is turned into a protein, and we're talking about the bit of DNA or bits of DNA which control when that information is accessed and transcribed, right? And that just little bit there, that's a huge field and realm of complexity, right? Some of which we'll touch on later on in the course. Okay. Yeah, get out of it. Here we go. Oh man, I've lost the chat box again. Oh, yeah. I figured out how to bring the chat box up without flapping around in it. That's a plus. Okay, so I don't need to know that. We've already covered that. This, however, if it goes on to it next, this is actually a very important piece of information, right? So this is, I'm pretty much sure I've mentioned this already. This is the equivalent of the Rosetta Stone for molecular biology, right? So every one of these, let's come back up to here, groups of 3 letters, right? So you see these letters that are being copied in this mRNA transcript, right? Let's assume that that's in a coding region, right? Rather than a untranslated region, right? UTR. These letters are grouped into words of 3 letter words, right? We don't have any, it's actually, if we were doing those Scrabble games on the phone, super easy, right? You wouldn't have any 7-letter words or anything. They're all 3-letter words. And those 3-letter words equal an amino acid. So one 3-letter word, one codon equals 1 amino acid. And only that 3-letter word can only ever equal that 1 amino acid, right? You can't get another amino acid put into the polypeptide at that position for that same codon, right? So, and that's really, let's kind of bump down to here a little bit. That's dictated by tRNAs, right? I'll come back to that codon table in a little bit, right? So tRNAs are this is what I mean by bouncing around a little bit back and forth. It's not necessarily a very linear process, right? So these tRNAs are the intermediates, right, between the information in the messenger RNA and the polypeptide sequence, the amino acid sequence of the protein, right? So once this is finished and it has a chance to fold or get wherever it needs to go, it will be able to do its job, right? So why does my chat box keep disappearing? Anyway, So these tRNAs are intermediates because they can only carry one amino acid. One tRNA, one amino acid, right? And in addition to that, at the end of the tRNA, we have what's called an anticodon, right? So if you remember back up here, come on, right? We have the base pairing between DNA and mRNA, right? To create this mRNA transcript. Down here, well, where did we go? We have base pairing again, right? Between RNA and RNA in this case. So we have base pairing between what's called the anticodon on the mRNA, sorry, and the actual codon, the information, the word on the mRNA, right? So this anticodon is the exact opposite of the codon. And so you can see here our start codon is an AUG, right? So that always encodes a methionine and only a methionine. And give me a second, I've got to show you a puppy, if only just to shut him up for a little bit. There you go. So hopefully you can now see what is causing all the bloody noise that's distracting me while I'm trying to teach this class. They are very, very cute. They are a complete pain in the posterior as well sometimes, particularly when they fight each other. I'm not sure what they are. They're mutts. I think the wrinkly one might be maybe Shar-Pei Terrier mix. I don't know what the other one is. Probably a bit of Beagle, bit of Terrier. We're fostering them, so they'll be gone soon, hopefully. Not hopefully because I want to get rid of them, but hopefully because then they'll be with their new families.
So your dog just had puppies?
Sorry, say again?
Your dog just had puppies?
No, we're actually so we have another dog, but she's been spayed. These two are fosters that we're looking after. That their mother was abandoned, and then I think before they could get to her, she was run over and killed. And so we had those from when they were 2 weeks old or 10 days old, something like that.
My dog just had puppies, 7 of them.
Wow, it's a lot of work. Wow,
They just started opening their eyes 2 days ago.
that's awesome. Wow, thank you for sharing that.
No problem. I just wanted to show puppies.
Yeah, totally. Well, if my other dog kind of finds her way down. She's a bit of a teenager. She isn't out of bed yet. I'll introduce you to her as well. All right, so back to the original plan. Hopefully they'll be a little bit quieter. It's just there's like this sea of squeaky toys and stuff that they're just, it's a little distracting. I might have to go up to my another room, so maybe another time. Anyway, focus, Matt. tRNAs, right? So they use the same properties that hold two DNA strands together, that hold a DNA and mRNA strand together, right? Those hydrogen bonds and shape, to bring in exactly the right amino acid for that word, for that codon, right? And so so each word is 3 different letters, which is, what's that, 3 to the power 4, 64 different combinations, I think. Come over here. And, 4 to the 3, sorry, get that the wrong way around as well. And what you'll see here is that each of those words encodes a specific amino acid. Now the flip side is different, okay? So we only have one codon equals one amino acid, right? But any given amino acid can be encoded by more than one codon, or more than one tRNA, essentially, right? And this is called degeneracy, right? So the genetic code is degenerate because there's a lot of redundancy here, right? So leucine is the champion of the hill, right? Because you can get a leucine amino acid in any given position where there are 6 different codons, right? So CUU, CUC, whatever, right? They each encode leucine, whereas there's only a couple of amino acids which are encoded only by one codon. That's the start codon, which is methionine. It's called the start codon because that's where translation starts, and every single amino acid, every single protein, as it's being synthesized at least, begins with methionine. Right? Tryptophan's the only other one which is encoded by one single codon, right? So I'll get to stop codons in a second. So the neat thing about this is that this essentially creates a buffer against mutation. So if you change, and we'll actually, this is one thing. They're super frisky this morning. This is, sorry, it's just one of the challenges of having puppies, I guess. This methionine, the start codon, is something that we'll be actually studying in lab. So this is the site of the mutation that makes yellow flies, right? So our fly lab, which is going to be a mix of doing some linkage experiments and then doing some molecular biology on the yellow gene, right? This is the target, or not the target, this is the site, sorry, of the mutation that causes flies to go yellow. And the reason is that you change any one of those letters, you no longer have a methionine, right? So if you change the G to an A, it's now isoleucine. If you change the A, at the beginning to a G, now you have valine, right? And so it just doesn't work. So for all the other amino acids, with the exception of tryptophan, you have a buffer, right? So for example, with serine, if you change the last letter, U to U, C, A, or G, does not matter, right? You still get a serine. And so that's a way of ensuring you still get the same amino acid sequence, right? You still get the same protein, even though mutations can randomly occur in a coding sequence, right? So it's kind of a buffer against mutation, right? Obviously, mutations will occur which will change the amino acid sequence, and therefore, you might get either a non-functional protein or one which functions a bit better or differently or whatever, who knows. So the only other oddity on here are these things called termination codons or stop codons, right? And just as you have to begin translation somewhere, you also have to stop it, right? You can't just keep going. It's just a waste, and you'll end up with all kinds of crazy stuff that you don't want. So at some point, you've got to be able to stop the process of translation, and that's what these codons are for. So every single coding region or reading frame or whatever you want to call it, in every single gene, I think in every single organism, can't think of any that don't actually. This is a universal thing. So this is a very, if you want to think about evolution, this is a very ancient piece of information that we're looking at here. This is basal to everything. So everything that is currently living's last common ancestor, right? Wherever that appeared, I don't know, a couple of billion years ago or whatever, used this codon table, right? This genetic code. Anyway, so that's just something that occurred to me as I was talking about it. So these termination codons, they don't have a tRNA that recognizes them. Instead, they have a little protein that comes in and essentially levers the ribosome off the mRNA, which stops translation, right? So wherever you get one of these, translation stops, like hitting a brick wall. And actually these, if you look through, for example, oh, here we go, tyrosine. If you have a codon that encodes tyrosine, and that last letter changes from a U or a C to an A or a G, now that protein stops. So if that tyrosine is halfway in, like if you have your reading frame, your genetic sequence, I guess, and the mRNA that's been translated, and there's a tyrosine in the middle, and in the DNA that's mutated to create a stop codon, that's how long your protein will be. It won't be the full length, it'll be truncated, which is usually catastrophic. It doesn't work anymore, right? So mutations that generate stop codons are almost without exception. I don't know what all the mutations in the world are, but I can't think of any where they would produce a protein that still functions, right? Very rarely, unless they're right at the very, very end. In fact, I even worked on a gene for 5 years, actually, funnily enough, that had a mutation 8 codons from the end of the transcript. And it was a null mutation, didn't work at all, no function. That protein didn't work. And it was only missing, sorry, I can't hold my fingers up very well, only missing 8 amino acids, right? So anyway.
So by definition, would a mutation be a protein where there's a stop codon before the end? Is that what you're...
No, really, mutations could be all kinds of things. That's a really good question. Actually, let's just jump ahead a little bit down to here. Actually, what's that one? So mutations are random, right? And it's totally random when they occur and where they occur. So, and what they are, right? I mean, some agents produce certain types of mutations more frequently than others, perhaps, but where they occur is totally random. And so most of our genome, 95% of our genome, is full of stuff that doesn't encode proteins, right? Only 3 to 5% of our genome encodes, has protein encoding sequences, right? And so if you think about just random chance or probability, those random mutations could occur equally randomly, right? Anywhere throughout our genome. And so the chance of a mutation actually landing in a protein coding region is actually fairly small, right? That's why most of our protein encoding genetic information is very, very similar to our cousins and nieces genetically, right? So like chimpanzees and great apes and things like that. We're very, very similar in terms of our protein encoding information, right? Now, if a mutation does occur in a protein coding region, right? So we go back up here. It could still be what they call silent or neutral, right? Has no effect, because if a mutation, let's say we've got a change from a C to an A in the third position of this codon here, has no effect, right? Even though the DNA sequence of that gene will be different from the DNA sequence of the same gene in another individual that doesn't have that mutation, the protein that's produced will be exactly the same. And if the protein is exactly the same, that's all we really care about, right? You know, it's going to do the same job, whether or not the information that encodes it is slightly different, right? Does that make sense?
Yes, it does. I'm just trying to figure out the reason to even have the stop codons. And
Oh, well, the stop codons, oh, I can see where you're coming from. Like, they seem to be inherently dangerous things to have, right?
yeah, it just seems like why, like
Totally.
what purpose do they serve, I guess.
So, hang on, give me a second. As they say, a picture is always worth 1,000 words. So the reason for having stop codons, actually, let's use red. Blue's a little staining up my board a little bit for some reason. So essentially, when you have a transcript, right, 5' to 3', sorry for the reflection on the window. I'll have to close the blinds in future. Right, so this is our mRNA. We need somewhere for ribosomes to assemble, right, before they continue the process. This is actually more in eukaryotes. But kind of the same principle applies in prokaryotes, but just for different reasons. And so we have a region. This is called our coding region. Or could also be called an open reading frame. There's lots of different words for it. This equals protein. Right, let's make sure you can actually see. Hang on, let me close the blinds. Gonna be a little bit darker in here, but you can still see me well enough. Right, that's a little bit better. Right, so this region here is what equals the protein, so you have to have a start codon here. That's what tells the ribosome this is where we start producing protein, right? Because we don't want to produce protein here because this doesn't code for protein. This is it's actually called an untranslated region, but you don't want to include the information here because you'll get a protein that's different. And the same applies over here, right? So this is another untranslated region, which you don't want to turn into protein. This is actually used a lot for regulation, like stability of the mRNA transcript, which again, we'll get to later on. And so you have to have a stop, because otherwise the ribosome is dumb as a box of bricks, right? It's not going to know where to stop unless you tell it to. And so if you don't have a stop codon, you'll continue on translating into this untranslated region, and that will produce a protein, right? Which instead of going from here to here, is going to keep going on into here. And this stuff you don't want because it's going to produce a protein that might not fold properly and might not function properly. So just as you have to be able to start translation somewhere, you know, a very specific point, you also have to stop it at a very specific point. Now, the flip side of that, as you've kind of what's prompted your question, is that if you have a stop codon anywhere in here through mutation, right? Let's say we have a stop here, then you get a truncated protein as a result, right? That's just the downsides of having this kind of function in the first place. Now, those stop codons, because they generate these mutations, they generate kind of null, they're null mutations, right? They don't work, the proteins. Those can still persist in the form of heterozygotes, right? Which we'll get to in Mendelian genetics later on. If they're in something critically important, then that organism may not survive. So therefore, this mutation will be selected against, right? So that's kind of like a kind of leading on to the bigger question of, you know, what happens once you get a mutation, right? And we'll talk about that in just a second. Did that help?
Yes, it did.
Yeah. So I wouldn't think of these as being, I mean, they are catastrophic if they do occur, but they don't necessarily occur very often, right? Otherwise, you know, we'd be just kind of gibbering balls of cells which don't function because we'd have huge amounts of mutations and nothing would work, right? So there's actually and another thing that we'll get to in DNA replication is there are a lot of very effective mechanisms that work to correct errors in DNA replication and mutations in general. But again, on the flip side, pardon me, we are the result of at least 100,000 years worth of mutations, right? That's why the way we are, right? We are essentially the mutational history of our forebears, right? And so we have the features and functions that we have as the result of mutations that were accumulated by distant ancestors in our evolutionary history. And those mutations, for whatever reason, right, I'm kind of bumping ahead. We'll get back to RNAs in a second. Essentially, those mutations, oh, here's a good question. Aha. I'll get to this chart in a little second. Where in your body, right, must a mutation occur for you to pass that mutation onto your offspring? Right. So if you go out, for example, if you go out in the sun, which is like almost all the time in San Antonio, and you're from more temperate climes and you get sunburn and you get mutations in your skin from the UV, will you pass those mutations on to your children? Question to the class. Yes or no? No. Oh, sorry. I didn't see the chat come up. Oops. Sorry. No, right? Because where do those mutations have to be for you to pass? Now, what did Anna say? Has to occur in the genome. That's kind of default, right? Because that's where we accumulate mutations in our genome. But what type of cell? What part of your body? Right. Think about what goes into the next generation. Yeah, it's got to be DNA in your nucleus. Nucleus of what cells? Right. So clearly, I don't pass on my skin cells unless I'm rubbing them on top of their heads to my children. Sex cells. Right. Exactly. Thank you, Jonathan. Germline or sex cells. Right. So only mutations that occur in your germline, your sex cells, your gametes, sperm or oocytes, depending on if you're male or female, only those mutations get passed on to the next generation, right? So let's say I have, well, I have 2 children, right? And I passed on a mutation to each of those just by random genetic chance. They both got the same mutation. how many copies of that mutation, say it's totally new, I generated that mutation, I should be really proud of myself, right? How many copies of that mutation are you going to have in the greater San Antonio area? It's going to be me and my 2 children. So there are going to be 3 individuals with that mutation out of how many people live in San Antonio? 1.2, 1.3 million, something like that, right? So that's not going to be a common mutation. What happens then is if that mutation confers some kind of benefit, right? I don't know what that might be. Less likely to get sunburned, maybe, right? Some kind of benefit that enhances your fitness, and there's an environmental pressure that selects for that benefit, right? So if, you know, protection against sunburn, you get that mutation and you're in Greenland, it's not going to do you any good, right? There's not going to, it isn't sunny up there, or at least you don't get sunburn very often, right? So the selection pressure has to be there from the environment, right? So obviously down here, lots of sun, that can protect you from other problems, right? Like skin cancer, right? And so over time, and we're not talking about time as in my children's lifespan, we're talking about their children and their children's children, their children's children, their children's children. You know, we're talking about hundreds, if not thousands of years, right? That mutation, which I produced, long since deceased, right? That frequency can increase if it confers a benefit, right? If it confers a fitness to that population. And that's how you see those mutations becoming kind of common or more fixed in the population, right? We're talking about time, lots and lots of time and appropriate pressure that kind of selects for that mutation as well, right? So mutations happen every time your cells reproduce, right? They happen every time, you know, you are exposed to an agent which can cause mutations, whether it's sunlight, gamma radiation, you know, alpha radiation, smoking, eating steaks. Sorry, Texans, supposedly that happens as well, right? So every time, but what the consequence of those mutations is varies hugely depending on where those mutations occur, in what cells, in what genes, if they're in genes, what kinds of mutations they cause in genes, and so on, right? And so that is, if it will change, quite neatly summarized here in this table. I'm not going to read through it because you can read just as well as I can. But essentially, you can classify mutations by a bunch of different ways, right? Is it something that's does it occur in your germline or does it occur in the somatic cells, right? Somatic being everything that is not contributing to the next generation, you know, so eyeballs, skin, liver, etc. Right? We can get back to, actually, this is what we were talking about earlier in terms of stop codons, right? What's its effect on translation, right? Could have absolutely no effect whatsoever, right? Could be catastrophic, right? It could be a nonsense mutation, right? Which will stop translation completely, right? It just depends on where it is and what the effect of that change is, right? Which we can talk about later on. Okay, so just to kind of bounce back and finish up this slide, it actually took a little bit longer than I expected, which is kind of neat. There's some good questions and puppies as well. Hopefully there's none of you minded the puppy show. So kind of bouncing back a little bit, just to kind of, again, put this worm in your ear, so to speak, is that just wanted to share that RNA molecules are really cool. Yeah, said it. They're really cool because they can do a lot of different things. Actually, I only got 3 minutes for a tangent here, so I better be quick. RNA is the only molecule that can be both a store of information, right, which is how we think about mRNA, for example, right, contains codons, which equals amino acids, therefore protein, and an enzyme, right? So RNA molecules can also have enzymatic activity. Those are called ribozymes. Let me just chat that. Actually, let's make sure that goes to everybody rather than just the last person. Ribo, let's see if I can spell it right. My typing isn't very good right now. Right, so ribosomal RNA, which is all the way back here. Come on. Ribosomal RNA is the enzyme that catalyzes the formation of peptide bonds, right, during translation. It's not the ribosome. The ribosome essentially provides structural context. The rRNA is what does the work, right? So RNAs can be both information and enzymatic activity. They can fold on themselves. They can form three-dimensional structures, right, which is pretty wild, right? They can base pair to themselves, right? And they can do all kinds of funky stuff. And the really cool thing about RNA, and this is actually it's called an RNA world hypothesis, right? Because RNA can both contain information and do stuff, right, in terms of catalysis, it's actually, a lot of people think, or it's a very powerful theory, that RNA was the first genetic molecule, right, the foundation of early life around 4 billion years ago, right, because DNA can't do that. DNA can store information, but it can't do anything, right? It has no enzymatic properties. So one theory is that RNA was actually the very first genetic molecule that evolved or appeared, whatever the word would be. And then later on, DNA appeared as a more stable kind of alternative to RNA, as a more stable store of information. So there you go. Isn't that cool? All comes from the fact that RNA can actually do two different things and DNA can't. So anyway, that kind of quite neatly has led us to the end of the lecture. There are also lots of there are other competing theories as to the origin of life, which kind of all overlap. It's a fun thing. RNA, you probably have RNA in chloroplasts. You also have DNA in chloroplasts as well, just the same as you do in mitochondria. And that's largely because we think those were ancient organisms that were kind of swallowed, I guess, by early eukaryotes, right? So chloroplasts were kind of like an ancient algae, mitochondria being ancient bacteria. But they also, because they have their own DNA genome, right, even though it's quite simple, they also produce RNA and proteins in chloroplasts and mitochondria too. They're pretty funky things. All right, so thank you very much, everybody. Super cool questions. Any questions from any of this stuff, because we're just kind of running out of time, please do hold on to those and bring them to Wednesday's class, and we can go over them at beginning class on Wednesday. All right, thanks for a great lecture, everybody. See you all on Wednesday.